Hi everybody, in Lecture 4 we will discuss some features of the NCBI BLAST search engine. The textbook chapter 4 is quite informative on the process of performing a BLAST search on the NCBI portal, so we will focus on certain features of the BLAST alignment. We'll discuss the values of results and the basis of their interpretation. First, we will focus on new features of BLAST that were developed after the textbook was published. The lecture will summarize a number of practical applications of BLAST, differences between databases and engines that you could use. We will also discuss the alignment strategies and will introduce the general principles and algorithms for multiple sequences alignment. BLAST was developed 25 years ago by a group of mathematicians and biologists at the National Institute of Health. Dr. Stephen Altschul is an MIT Maths PhD graduate who designed the basis for the algorithm. Warren Gish was a molecular biology graduate from UC Berkeley who developed the deterministic finite automation or DFA function for word heat recognition. Webb Miller was a mathematician who developed the algorithm for DNA sequence analysis, and Eugene Myers is also a mathematician who is credited for BLAST development. Finally, Dr. David Lippman is currently the NCBI director and started his bioinformatics activities by developing the sequence similarity algorithm, later on transformed into faster search engine and ultimately into BLAST. Their paper describing the BLAST, published in 1990, has over 40,000 citations, making it one of the most highly cited paper ever. Altogether, the discoveries by this group of scientists are incorporated in many relevant applications and software in bioinformatics. BLAST is a very versatile application that performs pairwise or multiple sequence alignment of a query sequence against a target or a number of databases. BLAST uses the heuristic approach based on Smith-Waterman algorithm that is an exploratory fast discovery interrogation resulting in an optimal alignment. The application finds the best local alignment and provides a statistical confidence value of the results. It could be used in almost any combinations of the molecules, such as DNA versus DNA, DNA versus proteins, and variations of thereof. BLAST is available as a standalone client, integrated in some commercial applications, and as a web-based application programming interface or API, creating a request-response message system. A number of specialized versions of BLAST are available on the NCBI portal, such as, for example, Primer BLAST, an application which allows the researchers to design primers based on the sequence ID or FASTA file, or to check the submitted primers for the product generated and the potential off-target effects. The list on this slide is an informative set of specialized BLAST services accessible via the link indicated at the bottom of the page. Some of the features and services are periodically modified, and the presented list is current as of June 2015. Some of the major advantages of BLAST are the speed of searching the databases, which is a major requirement for any bioinformatics tools, accuracy, availability of the engine on any browser connected to the internet, and the ability to modify the programming codes on which BLAST was developed. Application of BLAST include identification of orthologs and paralogs, discovery of new genes and proteins, variants and isoforms, investigating the coding sequences, and exploration of functional and structural similarities of targets. The BLAST steps are described in more details in the chapter 4 of the textbook. Major point to remember is that the order of indicated steps is conventional, as you can start the settings from any step. Basically, there is more than one way to skin a cat. It is, however, important to remember to recheck your settings before you push the button BLAST and start the search. One of the suggested tips that may be useful in using BLAST is to display the results in a new window. By default, the search engine results will open in the same window if you don't check the Show the Results button in the new window. Once you check it, the results will display in a new window or a new tab. By enabling this feature, your search window will have your settings and input queries preserved, so that if you would want to repeat your search with different parameters, you will not have to re-input your queries. The second identical box showed results in a new window is located within the algorithm parameters section. If you click the plus sign, you will see it next to the second button, BLAST. You don't have to check both radio buttons, because checking on either top or lower section will enable it across the search engine. The following several slides are just snapshots with some options explained. One of the main points to remember is that the search query may have only the FASTA format or IFSEQ or GI IDs. GI stands for Gene ID. Once you enter the RefSeq ID or GI ID, the query window will be populated with the FASTA sequence of the molecule, the ID of which you enter it. If you mistype the ID, it will give you an error, something like no sequence found. BLAST can be started from almost any NCBI databases where the sequences are displayed. You can select a region within a genome and blast it from there without getting into any specific databases or inputting your query. If you do it from within a sequence displayed on your screen, BLAST will automatically detect whether it is a nucleotide or protein and will suggest applicable engines. If you start your search from within the BLAST portal directly, you will have to select the appropriate engine to search for your sequence. BLAST-N is the main nucleotide alignment engine, BLAST-P is for protein input, and the additional options can translate the sequences from one type to another and align them correspondingly. BLAST algorithm can be specified within the settings prior to starting the search. Different types will generate somewhat different reports depending on what features you have enabled in the parameters section. For the purpose of this lecture, we will use only the default features, but it is useful to know that many other options are available, and I would recommend investigating them on your own. Such specialized features may be more appropriate for specific research questions in which you would want to find details on cross-species alignments, for example, or to find the similarity around specific motifs. The NCBI website has many guidelines and clarifications of the features, which are constantly updated. It is probably a good idea to check the help files periodically if you plan to perform some advanced searches. An alternative of running the BLAST or inputting the sequence is to initiate the search from almost any page on the NCBI portal by clicking on the Run BLAST link, particularly when you're looking at a gene or molecule within a specific database. In case that your input is a nucleotide sequence and you are trying to run a protein BLAST alignment, you will get an error as shown on this slide. Make sure that you select the appropriate program for your input query. It also could be helpful if you know the scientific name of the species that you are investigating. If your samples come from such species, you will probably know their scientific name. Once you start typing the common name in the space for species, a list of databases that contain this common name will appear in the drop-down menu, and you will be able to select the proper database. However, before doing a BLAST alignment, look up the official scientific name so that you can limit your search to the targeted species. The optional parameters are designed to help you in selection of the proper alignment algorithm, scoring parameters, and filtering or masking of your results. One of the most useful features of the NCBI portal is the question mark link against the majority of options and conditions. Your results may display in a new window if this option is selected. It might take from seconds to several minutes to generate the reports. It depends on the type of molecules, its size, and the parameters that you have selected. However, it also depends on the number of users that are performing the BLAST simultaneously. Usually during some NCBI workshops and demonstrations where a number of users are performing a similar search, the engine is slowed down. However, during the normal work hours, it is not likely that many researchers will perform the search on the same molecule unless an important discovery on some genes or proteins have just been published and publicized in many scientific journals. The report will display your sequence of interest and the alignments of top hits, along with the statistical data such as max score, total score, coverage of your query, the e-value, the percent identity, and the accession numbers of homologs. You can change the report to display the taxonomy and to get access to other features available on the NCBI Blast. Basically, the NCBI portal is filled with links from one place to another, and you can make rounds from page to page without having to change your molecule of interest. Sometimes it feels like you are traveling a labyrinth and that you have already been on this page. So it might take a while to get more familiar with the features and shortcuts for a more productive search. One of the most important statistical indicators on the BLAST report is the e-value, which is the number of hits per databases that you would expect to get by chance. Again, the lower is the e number, the less chances are that your findings are due by chance. As mentioned in the previous lecture, the number cannot be negative, and the minimum threshold that you would want to use is e at -4, meaning that there are 4 zeros after the integer and that your value will be less than 0.0001. If your e-value is larger than this, the similarity of your sequences are most likely questionable. Here's an example of how the Blossom 62 matrix is applied in calculations of the local alignment score. The perfect match of lysine k = k is rewarding +5 points. The substitution of k for e is still rewarding, but only 1 point, while the q for f is penalized -3 points. The gap score was calculated based on the gap penalty of 11.1 that was set up in the alignment advanced features, and the total penalty for this region of gaps is -14 points. Similar calculations are performed all along the alignments, and the resulting sum of these numbers is the max score indicated in BLAST report. On this slide, the e-value is much lower, and the principles of calculations are similar to the ones indicated on the previous slide. The multiple sequence alignment, or MSA, principles are similar to the pairwise alignment in the sense that it calculates the best alignment options between 3 or more molecules and reports a score of similarity. The main purpose of the MSA is to identify the molecules that have conserved regions. The MSA was used in computational prediction of the protein's functions based on conserved domains, and it is the basis of assumptions that proteins with similar domain have similar functions. The biological significance of MSA is in understanding how the 3D structure is formed, folding of proteins, grouping of molecules into classes such as immunoglobulins, receptors. Some specific amino acids contributed to the formation of either alpha helices or beta sheets. Also, based on the MSA, it was deducted that some particular amino acids and nucleotides are prone to mutations such as insertions and deletions. MSA is used to assess the level of conservation of protein domains and their secondary and tertiary structures. It could also be used in BLAST by aligning more than one protein. MSA is also used in GWAS SNP analysis. When you search a query against multiple databases, you in fact are performing an MSA. It is also important in identification of the regulatory regions of genes, as usually such regions have high similarity in their sequences. When you look at the MSA report, you would see that the homologous residues appear in columns, and such molecules are homologous from the evolutionary point of view and as structural units. It is important to remember that MSA can align any sequences, but it does not mean that the sequences are homologous if the stats are not significant and the scores are low. The MSA uses the rewarding and penalization matrices similar to the pairwise alignment. The clustral W and other algorithms are using combinations of matrices and alignment strategies to detect the homologous sequences. The process is iterative, meaning that the interrogation is repeated constantly to detect the optimal alignments. Some of the approaches are using variations of algorithms and settings, such as fast Fourier transformation in MAFT and known 3D structures in espresso approaches. If you search the homologs in homologene database, you will see the conserved domains which serve the basis of MSA and pairwise alignments. The homology database is giving you the approximate similarity of proteins and DNA among different species that you could extend by clicking on the BLAST links. Finally, you could zoom into conserved domains and you will see the topographical details of those domains' locations between species. This slide shows a multiple sequence alignment of PCSK9 proteins in different species. As you can see, the overall similarity is about the same, the differences being in single digits between different approaches and algorithms. Usually the researchers are using the methods that are best understood to them and that is customary accepted for particular field of investigations. The small arrow displays the conserved histidine residues, the location of which may differ in dependence on the type of alignment and algorithm used in MSA. For example, in cluster W alignment, the histidine conserved regions are located at the position 558 and 593 of the above alignment. Similar to the muscle alignment, the same positions for histidine, and the global alignment used by Genius also indicates the same positions. However, in MAFT alignment, the positions of the histidine residue are at 568 and 601, despite the overall comparable statistic of MSA. In summary, the MSA is more difficult to perform and it requires more computational power. The results may and will vary between types of approaches, and as any types of alignment, it is the best or optimal alignment, which could be done in many ways, no correct or incorrect alignment. The MSA is at the basis of the evolutionary research and building the phylogeny trees of homology. This concludes the lecture 4. Examples of the NCBI and Genius alignment will be provided as separate short movies.